Hello friends, so welcome to this next video on of the series on genomics interpretation of clinical data. So here in this video we will see briefly about the expressivity and penetrance. So what is penetrance? Penetrance refers to the probability of a gene or trait being expressed, and expressivity on the other hand refers to variation in phenotypic expression when an allele is penetrant. So, penetrance in some cases, despite the presence of dominant allele, a phenotype may not be present. A dominant allele. So here we have taken one example of polydactyly in humans. Polydactyly is presence of extra finger or toes. A dominant allele produces polydactyly in humans, but not all humans, while the allele display the extra digits. Complete penetrance means the genes or genes for a trait are expressed in all the population who have the genes. Incomplete or reduced penetrance means genetic trait is expressed in only part of the population. The penetrance of expression may also change in different age groups of a population. Reduced penetrance probability results from combination of genetic, environmental and lifestyle factors, many of which are unknown. The phenomenon can make it challenging for genetic professionals to interpret the person's medical history and predict the risk of passing a genetic condition to future generations. Now, expressivity is a variation of phenotypic expression. So again we will consider the example of polydactyly, where an extra digit may occur on one or more appendages. The digit can be full size or just a stub. Hence the allele has reduced penetrance as well as variable expressivity. Variable expressivity refers to the range of signs and symptoms that can occur in different people with same genetic condition. As with reduced penetrance, variable expressivity is probably caused by a combination of genetic, environmental, lifestyle factors, most of which have not been identified. If a genetic condition is highly variable in signs and symptoms, again it may be challenging to diagnosis. Now we will see some terms related with the penetrance. Pseudo-incomplete penetrance is the definition of non-penetrance depends upon the accuracy of the clinical phenotypic analysis. Non-penetrance is inaccurate because the clinical examination is incomplete or the symptoms have not yet appeared at the time of examination. Now we will see what are the factors that affect this penetrance. First is permutation. The incomplete penetrance in this case is due to the existence of intermediate state of mutation with no clinical expression. The mutation may change from one generation to another. Therefore, a normal individual with pre-mutation may have several affected children in whom the mutation is fully penetrant. The next factor which affects the penetrance is sex limitation. For some mutations, the clinical symptoms are present only in one sex. Sex limitation is often seen in disorders in which the expression of the disease is limited to organs found in only one of the sexes, such as testes and ovaries. The next factor that affects penetrance is influence of alleles. In dominant disorder, the clinical phenotype is present in heterozygous. Either a polymorphism or a mutation of the allele in trans may lead to the incomplete penetrance of the dominant mutation. Influence of the modifier gene is another factor which affects the penetrance. The presence of a mutation or polymorphism at a locus different from the one causing the disease may alter the effect of mutation and prevents its clinical manifestation. The next factor that affects the penetrance is diagenic inheritance. The term diagenic inheritance was proposed to describe a situation in which the clinical expression depends upon the presence of two mutations in two different genes. And finally, imprinting is the factor which affects the penetrance. Both parental genomes are essential for normal growth and development. A mutation in one of these imprinted genes will express only if present on the chromosome from the parents who transmit the active copy of the gene. So these are the factors which affect the penetrance. Now we will see the expressivity. Expressivity is the phenotypic difference. Mutation in the same gene may cause different clinical symptoms and two different groups of expressivity may be distinguished. The same molecular genotype may be present in either the severity of the same clinical phenotype or differences in clinical presentation. Now we will see what is pseudo differences in expressivity. Homozygotization of a dominant mutation may give the impression of difference in expressivity because the phenotype of the homozygote is often more severe than that of heterozygote. In family which an affected individual presents with severe form of a dominant disease, the possibility of homozygotization may be effaced if both the parents present symptoms. However, the possibility may not be evident either because of the incomplete penetrance in one of the parents or uniparental disomy in the child. Now we will see the effect of different factors on the expressivity. First is influence of the allele in trans. Either a polymorphism or mutation in the allele in trans may lead to the difference in the expressivity of a dominant mutation. Second is influence of polymorphism present in cis. Several polymorphism mild mutations have been reported that if they are combined in cis, they lead to severe phenotype. Next is influence of another gene. The clinical presentation of monogenic disorder may depend on the presence of mutation in another gene. Next factor is anticipation. Anticipation is a term used to describe transmission of a dominant disorder with an earlier onset and more severe manifestation from one generation to the next. Next factor in the expressivity is influence of the sex. Clinical presentation of a monogenic disorder may depend on the sex of the affected individual. In some cases, a particular clinical symptoms may be present in only one sex. The next factor which affects the expressivity is somatic mosaicism. The presence of mutation as a somatic and germinal mosaicism often leads to a milder phenotype in the affected individual than his children. The affected children present a more severe form of disorder because the mutation is present in all their cells. The last is environmental factor. In many metabolic diseases, the clinical presentation may depend upon the quantity of substrate in the diet. So these are some of the factors that affect the expressivity. Now we will see the differences in the clinical presentation of expressivity. Molecular heterogeneity. In some cases, the differences in clinical presentation are secondary to molecular heterogeneity. Some mutations are responsible for a more severe phenotype, whereas other mutation leads to milder phenotype. Next is influence of a polymorphism in cis. A dominant mutation may lead to different phenotypes depending upon the type of polymorphism present in the same gene in cis. Imprinting. According to the parent who transmits the mutation, a different clinical presentation may be observed due to imprinting. Finally, somatic mosaicism. The clinical syndrome may be different when the same mutation is present in somatic mosaicism or in all the body cells. So this was all about the expressivity and penetrance. So in the next lecture we will see about the introduction to genomics and central dogma. Thank you and keep following for the complete series on clinical interpretation of genomic data. Thank you.